Hiya, kids! Thanks for tuning in! The late 1900s was pretty heated, with lots of intense competitions between scientists such as Watson, Venter, and Sulston, and organizations such as the NIH, Celera, and the Consortium. To them, DNA sequencing is life. It was considered pointless to worry about to others, but they still persevered and did so.
Hello, I'm Dr. Sanger, and thank you for listening. So, after concluding my study, I found that to sequence the DNA, you must first separate the DNA into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA. Well, it's done. I, Dr. Sanger, have decoded the genes of virus X174. 5,400 bases. 5,000 nucleotides. What am I going to do with my life now?
Yes, we finally did it. We automated the DNA sequencing process. These machines, the automated DNA sequencer, DNA synthesizers, peptide sequencers, and an inkjet printer for constructing DNA arrays. Finally, we have shortened the DNA sequencing time.
And now it's in color. Order one now.
Sorry, I'm not interested in it. Bye.
Well, I guess I could use it to see how radioactivity hurts DNA. I'll take it!
And so, the Department of Energy opened the first human genome project for 7 years and a $1 billion effort in Los Alamos, which was across from the site of the Manhattan Project.
Look at them. iPhones, all that money. Wait, what if I made my own?
And that is how the NIH entered the race to decode the first human genome, with Dr. James Watson as their leader.
My word is the iron fist around here. What I say goes. Capiche?
Yes, Dr. Watson. Of course we will. You are a very revered scientist, no doubt.
That's what I like to hear.
Oh God, it's the Golden Girl.
Dr. Watson may come join. I don't see the benefit in reducing your agree that I can't believe the government is spending so much on this pointless project. It's going to be centuries before we can even use this data. Not only that, we're wasting our time and brain power on this pointless thing when we could be doing something that actually matters.
This is a PSA, public science announcement. You all are too weak, too afraid, and too thick-headed to travel into the new frontier of DNA that's waiting to be explored. You wait. I will pave the path for science, and that is a fact. Now, let me tell you, sequencing is the next step for science.
I signed up for the NIH. So my logic was pretty convincing. And you must consider that this important process takes time to yield results. So don't lose faith. Those results will be worth so much more in the long run. Bravo! Here you go.
And everything seemed all good and well. That is, until...
How dare you?
Wait, what?
You money sucker. I can't believe that you want to patent a gene. All of our scientific hard work, you're just throwing it away for money?
What's wrong with patenting a simple gene?
The business, you PH of zero. Are you kidding me? All of the scientists are now going to have to pay a toll whenever we have to study that gene just because you want to make some money.
Alright. To be honest, I don't even know what a gene sequence is, alright? Please, let's just work it out, let's talk it out.
You crossed the Rubicon, my friend. There's no turning back. of your policies of patenting, which was sporadic and stuff.
Well, he has to go, doesn't he? For what?
Watson, coming. Yeah, you called?
We're fine.
Whatever. I'm resigning anyway. I'm gonna be the one who ends our partnership.
And that is how the partnership between Watson and the NIH ended. But, as one legend walked out, another legend walked in. This is Venter's story.
You know, since I was young, I was a bad kid. I raced planes with my bike on the runways. I boycotted spelling tests. And my high school sweetheart's dad held a gun to my head because I had a so-called overactive Y chromosome. However, I've always wanted to have a good life and be in some top position, but I basically ruined my chances since I got F's in school. But my drive for that dream went on, and I decided to join the MASH hospital in Vietnam. I felt disgusted at the waste of lives, and I had no life purpose then, so I decided to commit suicide. As expected, I had to take the most dramatic route to go, aka drowning in the South China Sea. But when I got out there and the sharks and snakes started nudging me for food, I thought, what the fuck am I doing? And swam back to shore. My life now had a new purpose, medical research. I got my PhD in physiology, and now I'm here, only because my friend gave me this quick method to not hand-sequence genes by looking at mRNA. Anyway, this was too much talk. Let's get working.
And so, Venter got started with work at the NIH. His process of doing the...
Okay, NIH, I have a great idea.
Oh, okay, go, just go through it. It'll take about 3 years, though.
Hell no! I ain't doing that. I'll just take a shortcut.
Venter, that's just cheating. It's like taking a helicopter halfway up Mount Everest. You're not doing the real foundation work.
So what? As long as I get results. You just need to get them.
You know what? I'm done with you. You're too hard to work with. Alliance over.
And that is how Venter lost all his scientist friends. And in 17 years, he was so done with the bureaucracy that he resigned and joined a new hybrid program, Tiger.
I'm so happy you decided to join us, Venter.
You aren't like that NIH who's just a bureaucracy. You are dedicated to pure science, and I will build you up to be the topmost program in the world. Although, you do patent genes, and your money comes from healthcare corporations. And whatever, you'll work. Now, we need to get my 30 staff members from NIH. President. Well, now that we have everyone, I would like to announce the staff and the start of our whole genome shotgun sequencing.
Okay. You're too hasty, my friend. Our company's strategy is to first make a meticulous map of each chromosome, divide the chromosomes into segments and send them into separate divisions. We would then shotgun each section into tiny bits and sequence them. A very methodical, accurate process.
Okay, you oldies go and do your methodical ways while the real men do the real work. All we need to do is shotgun them all at once. It will be easy and almost fully correct. I don't know why you didn't give me the funding for it, but you're lost.
And he did it. 2 genomes were coded by Venter in much less time than it took the NIH to solve 1.
Check out my swag sign. It's definitely characterized.
Oh my gosh, guys. I can't believe we get to see him in person, even though it's like behind glass, whatever, that's totally fine. OMG, Dr. Venter. Oh my gosh.
Oh yeah. Finally getting the attention I deserve. And you know what? My team will be the fastest to figure out how to combine multiple genes from living women. That is, that the government is challenging. You know what? I'm gonna make a company for it.
Oh my gosh.
And that was how Celera was born.
created by Venture exactly? And first of all, what even is your name?
To be honest, I don't really have a name yet, but I was made to destroy the International Consortium. We will become the strongest scientific company in the world.
Okay, so what systems do you have right now, nameless, created by Venture Association?
Well, I have hundreds of the state-of-the-art sequencers, as well as the world's largest non-military supercomputer to process data, built by my creators. I truly am the most strongest company in the world.
Alrighty then, thanks for coming today. Ow.
Hey, I've got an idea.
What?
Let's challenge the International Consortium to sequence mice as the last of the valuable work they can do.
Awesome.
I just immortalized them.
Aww. Oh god. Venture just challenged the International Consortium to sequence mice DNA. He is the Hitler of DNA sequencing. I'm so done. He's truly a tyrannical beast.
Fuck that.
What do you want?
Okay, so I just found out about Venture's achievements, and I don't know why I came over here, to be honest. Okay, I just needed you to talk to you about this. You know, I'm getting scared for your human genome project. Venture may just surpass you even though you got a head start. You only have 4% of the human DNA. So, could you please consider coming back to...
No, there's no turning back.
Okay, no, I don't mean any of this. You understand that I had to do what I did, right? Okay then. I'll see myself out. Goodbye.
I'm leaving the consortium anyway. It's too hard to work without you.
Thank you all for coming today. I would like to announce our new head, Francis Collins.
Yeah, thanks. Let's skip all the formalities and speeches and stuff and just get to work.
How in the hell am I going to get Celery to love me?
Uh,
That infernal Vector is so overprotective. He won't even let me talk to her.
well, you should prove to him that you're better than he thinks you are.
A better way than an arms race against Venture for the love of Celery and the best equipment.
Are you sure you can face Venter?
Yes. Ha! What perfect timing. I challenge you to an arms race for the best sequencing equipment. I will get Celery. You just watch.
This competition between Venture and Sulston got pretty heated, so bad that there were even death threats. But Sulston is one stubborn guy and kept fighting for Celery.
He's definitely not going to win with this new method I've got called blitzkrieg sequencing.
Venture, are you really sure that this is worth it? All the scientists already hate you, especially for going away. But now they're really criticizing you for not knowing this function.
Anything for you, Cells. You just wait. I'm going to save you from that creepy stalker Sulston. And they'll always use my methods later anyways.
Okay.
Well, he ended up making that blitzkrieg sequencing, and everyone started using it. However, the competition for Celery got interrupted.
Now, I know that all the scientists have been having an exciting race for the human genome, but you can't keep away information from the rest of us. The human genome belongs to the people. So now, you'll immediately share sequence information. That's all. God bless America.
Oh God, what's gonna happen to Celera? To the whole competition? Oh my God, Celera, we're broke.
Well, we're not entirely broke.
We just lost $6 billion in stock value. We can make it up.
We're broke. There's no way we can win this.
There's only one way to solve this.
We know that all the people of America have been following the human genome competition pretty closely. And the scientists have been motivated to work by your viewership too. But the big moment is here. Here are the results. God bless America.
We have officially agreed to end this competition in a draw. Thank you, Celery, for coming today. Now I have one question and one question only. Will you please address the audience and tell them, did you poach consortium data?
Yes.
I mean, there's always times in our lives that we regret, but of all people, you? Okay, now we're going to welcome to our show the consortium leader, Sulston. Hi, Sulston. So, how do you take this information of Celery poaching consortium data?
You know, what was in the past is in the past. Why bring it up now? I mean, every scientist agrees that her work is better. It was for the advancement of science.
Interesting point of view. Well, that's all, folks. Stay tuned next for our exclusive special, Love and DNA, a race to sequence the human genome.
A few months later,
You told me that this was free information, that I would never get hurt because of this.
I had no idea they'd find you. It was only small information, not that important, not that bad.
No, you lied to me. When I called there, you didn't even attempt to save me from that interview, from the shame that I felt. And you had a promise to always protect me, but when it mattered the most, you broke it. I hate you.
But I worked so hard for it.
No, you're fired. Go.
Sully, I feel bad about the whole fight between Celery and Vector, and how it's affected Celery. I mean, she lost her motivation without him, and to be honest, that's the only reason we won.
I know what you mean. I'm happy that we won. We finally decoded the human genome fully. This is what we dreamed for. Let's go.
What? You did what?
Hello, Vinter. Welcome to our show today. I've heard from various sources that you're the anonymous donor. Is this true? Yes. Joke's on you, everyone. You just decoded my own genome. Wow. Just wow. I have no words for this. This is classic Vinter, no?
So that is why I thought I should make the TCAG, or the Center for the Advancements of the Genomics Project. By sequencing my DNA, we can finally have a map of our genomes and about the essence of our lives.
More like your life. Oh, watch it!
Anyways, this is $100 billion spent in 4 years, so you better give a big welcome to the TCAG.
I hope we can work well together.
I hope we can work well together too.
Okay, I'm still waiting for my revenge. And what better way than to rival your DNA company with my DNA project, Jen? Basically, we're sequencing my DNA and it's going to be better than you, as always.
Bring it on. Just gives me another chance to prove that I will be the one to win over my competitor. Please.
but there really wasn't any results. No cures have been found in the Human Genome Project since 2000, although sequencing DNA has led to more precise biological clocks, revealed that animals harbor huge stretches of viral DNA, help scientists to reconstruct the origins and evolutions of the branches of human life and to trace global migration, confirm how few genes humans have, around 26,000, and led to the acceptance by scientists that humans are not special because they possess special DNA, but because they regulate and splice DNA in a special way. Even though they decoded the human genome, it makes all the difference between reading a genome and understanding it, as the ACGT sequence can be circumscribed by extra genetic factors, including epigenetic factors. However, in their defense, finding mutations to fix is very difficult, and most common diseases have more than a few mutated genes associated with them. So it's very hard to design a drug that targets so many genes. But when you think about it, all healthy bodies resemble each other, while each unhealthy body is unhealthy in its own way, as quoted from the Violinist's thumb. So, who knows if a disease is really just similar symptoms but different causes?